Welcome, everyone. Today, we are diving into a truly fascinating topic in human physiology, the hormonal control of gastrointestinal enzymes. Have you ever wondered how your stomach knows exactly when to start digesting and when to stop? It is not magic. It is a highly sophisticated chemical communication system. Let us explore how this life-sustaining system works. To understand this system, imagine you are running a massive, highly efficient factory assembly line. This line represents your gastrointestinal tract. If the workers, which are your digestive enzymes, are working at full speed when there is no food on the belt, the factory wastes a massive amount of energy and might even damage its own machinery. Conversely, if a huge shipment of food arrives and the workers are asleep, the food just rots. To prevent this chaos, your body uses a highly advanced text messaging system. These chemical text messages are called hormones, and they ensure your body only digests food when food is actually present. Now that we understand the factory analogy, let us look at the actual biological sensors. Hormonal control refers to a highly regulated endocrine system. Lining your stomach and small intestine are specialized cells known as enteroendocrine cells. You can think of these as the quality control inspectors of our factory. They act as chemical sensors that detect the exact type of food you have just eaten. When these cells detect specific nutrients, like fats, proteins, or acids, they immediately secrete specific hormones directly into your bloodstream. This is the crucial first step in dictating the timing, volume, and composition of your digestive juices. Building on those chemical sensors, let us identify the specific messengers they release. There are three primary hormones involved in this gastrointestinal process: gastrin, secretin, and cholecystokinin, which we often abbreviate as CCK. Once released into the blood by the enteroendocrine cells, these hormones travel to target organs like the stomach, the pancreas, and the gallbladder. They do not just float around randomly. They bind to specific receptors on these organs. This binding action is the exact trigger that releases the specific enzymes needed to break down the exact meal you just consumed. You might wonder why the body goes through all this trouble instead of just releasing enzymes constantly. The primary reason is cellular protection and energy conservation. Digestive enzymes are incredibly powerful and biologically expensive to manufacture. If your stomach and pancreas secreted these protein-destroying enzymes continuously when empty, the enzymes would literally begin to digest your own organs. This would lead to severe ulcers and catastrophic internal damage. By keeping the enzymes locked away until the exact moment they are needed, the hormonal control system protects the delicate lining of your digestive tract while saving massive amounts of cellular energy. Beyond just protecting the organs, this precise timing is necessary because of the chemistry of digestion. Different foods require completely different chemical environments to break down properly. For example, digesting a heavy protein like a steak requires a highly acidic environment. On the other hand, digesting fats requires a neutral environment mixed with bile. If the body just released everything at once, the chemicals would neutralize each other and digestion would fail. The hormonal control system ensures that the right enzymes are released in the exact right amounts, maximizing nutrient absorption from whatever specific meal you just ate. Now that we understand the biological need for this system, let us look at how physiologists model it mathematically. The relationship between hormone concentration and enzyme secretion is not a straight line. It is a saturation curve. This happens because the target organ only has a limited number of hormone receptors. We describe this using a modified receptor binding equation. In this formula, S represents the actual secretion rate of the enzyme. S max stands for the absolute maximum secretion rate the organ can achieve. C represents the current concentration of the hormone circulating in the blood. Finally, Kd is the dissociation constant, which is the exact hormone concentration needed to reach half of the maximum secretion rate. As hormone concentration increases, secretion increases, but it eventually levels off at the maximum limit. Let us apply this formula to a real-world physiological scenario. Suppose a patient eats a heavy meal, causing their blood CCK concentration to rise to 4 nanomoles per liter. Their pancreas has a maximum secretion rate of 120 milligrams per minute, and the dissociation constant is 2 nanomoles per liter. We want to find the current secretion rate. First, we substitute our given values into the equation. We replace S max with 120, C with 4, and Kd with 2. Next, we simplify the denominator by adding 2 and 4 to get 6. Dividing 4 by 6 gives us approximately 0.667, meaning the pancreas is operating at 2/3 capacity. Finally, multiplying 120 by 0.667 gives us our final answer. The pancreas is actively pumping out 80 milligrams of digestive enzymes every minute. Having successfully calculated the secretion rate, let us look at the system from the opposite direction. Endocrinologists often need to find the required hormone concentration for a specific target secretion. Imagine a patient needs a stomach secretion rate of 90 milligrams per minute. The maximum rate is 100, and the sensitivity constant is 1.5. We need to solve for the unknown concentration, C. We begin by plugging our known physiological target values into the standard secretion equation. To start isolating our unknown variable, we divide both sides of the equation by the maximum rate of 100. This gives us 0.9 on the left side. Physiologically, this tells us we are trying to achieve exactly 90% of the stomach's maximum digestive capacity. This sets up our equation for the next algebraic steps. Continuing from our previous setup, our next mathematical goal is to eliminate the fraction entirely to make solving for the variable easier. We do this by multiplying both sides of the equation by the entire denominator, which is the expression 1.5 plus c. This crucial step cancels out the bottom of the fraction on the right side and brings our unknown variable up to the main line of the equation. Now that the fraction is gone, we must expand the left side. We distribute the decimal 0.9 across both terms inside the parentheses. Multiplying 0.9 by the constant 1.5 gives us a value of 1.35. Similarly, multiplying 0.9 by our variable c gives us the term 0.9c. We are now very close to isolating the hormone concentration. Now for the final steps of our derivation. At this stage, we have the variable c on both sides of the equation, so we need to group them together. We accomplish this by subtracting the term 0.9c from both sides. Since the right side is essentially 1.0c, subtracting 0.9c leaves us with exactly 0.1c remaining on the right side. In our final mathematical step, we divide both sides of the equation by 0.1 to completely isolate the variable c. This simple division gives us a final calculated answer of 1.5 picomoles per liter. This specific number tells the diagnosing physician that the patient's enteroendocrine cells must release enough gastrin to reach this exact blood concentration in order to successfully digest their meal. Having proven the math, let us visualize this biological reality. Here you are looking at the hormone secretion saturation curve. On the horizontal axis, we have the hormone concentration in the blood. On the vertical axis, we have the resulting enzyme secretion rate. Notice how the curve starts at 0 and rises very steeply at first. This shows that the organ is highly responsive to initial hormone signals. However, as the hormone concentration continues to increase, the curve flattens out, approaching that top dashed line labeled S max. This represents the absolute physical limit of the organ, when every single receptor is bound. Also, look at the point labeled KD on the horizontal axis. This is the exact concentration where the organ reaches exactly half of its maximum capacity. We have covered a lot of ground today, so let us summarize the key takeaways. First, the hormonal control of gastrointestinal enzymes is a beautifully orchestrated system of chemical communication. By using hormones like gastrin, secretin, and CCK, your digestive tract precisely matches its enzyme output to the specific size and composition of every meal. Second, we saw mathematically how this dynamic, receptor-based system operates on a saturation curve, preventing the dangerous autodigestion of your internal organs. Finally, this entire system exists to conserve valuable cellular energy while guaranteeing that you extract the maximum possible nutrition from your food exactly when you need it. You now have a solid understanding of this vital, life-sustaining system.